everyone, today we are going to learn atomic force microscopy. In today's lecture, we will be learning about scanning probe microscopy. Under the scanning probe microscopy, we will be learning about the scanning tunneling microscopy and atomic force microscopy in detail. At the end of the lecture, we will also have a practical demonstration to understand the atomic force microscopy in detail. Let us see what is scanning probe microscopy. Scanning probe microscopy is a high resolution microscopy used to study surfaces at the atomic and nanometer scale. By using this scanning probe microscopy, we can get high resolution image and we can also get the quantitative data, and we can do surface specific analysis, and we can use any kind of sample. That is the advantage of this scanning probe microscopy. Under the scanning probe microscopy, there are several types of microscopy, and in today's lecture we are going to learn about scanning tunneling microscopy and atomic force microscopy in detail. Let us see what is scanning tunneling microscope. This scanning tunneling microscope STM was invented in 1981, and the STM is the first instrument to image surfaces at the atomic level. So, it was a breakthrough in the nanotechnology field, and for this research in 1986 these two scientists got the Nobel Prize. Let us see the working principle of scanning tunneling microscopy. Scanning tunneling microscope relied on the tunneling effect. That means this microscope is mainly working on the basis of tunneling current. It is going to measure the tunneling current between the sample and tip. The principle of this STM is, when you apply the small voltage, electrons can tunnel through a vacuum gap between the sharp metallic tip and a conductive sample, and due to the overlapping of their respective electron clouds, the tunneling current is produced. The tunneling current will be measured, and that will give the final image. So, this red color is the your tip and the blue color is the sample. The STM is going to measure the tunneling current between the tip and sample. The change in the distance between the tip and surface while scanning changes the tunneling current, and that helps in determining the topography of the sample. For example, you are having a sample like this, and when the tip approaches here, the tunneling current is different, and when the tip approaches here, the tunneling current will be different. So, it is going to measure the tunneling current and it is going to give the final image. But one major drawback or challenge in the STM is it requires a conductive sample and vacuum environment, which is one of the problem in the using STM. So, that is the reason atomic force microscopy was developed to overcome the limitations of STM. The first atomic force microscope was built by the same two scientists along with Professor Kelvin, and the advantage of this AFM is it can be useful for studying even the non-conductive materials like polymers, ceramics and biological samples. So, that is the advantage, and here we do not need any vacuum condition also, that is another advantage. So, this overcome the drawbacks of STM. Let us learn about AFM more in detail. AFM provides 3D topographical map of a sample by measuring the interaction forces between a sharp probe tip and the sample surface. So, this is going to measure the interaction forces between the tip and sample. So, that is why it is called as AFM. It is measuring the interaction force, that is why it is called as atomic force microscope. Whereas, in case of STM, that is measuring the tunneling current, so it is called as scanning tunneling microscope. This AFM can determine the surface features with sub-nanometer resolution. That is the advantage of this AFM, and this AFM is mainly depends on the mechanical interaction. As I told earlier, it is mainly depends on the mechanical interaction such as van der Waals or electrostatic forces between the probe tip and the sample surface. Let us see the parts of AFM. The first one is cantilever. The cantilever is a thin flexible material with a low force constant, and the tip is mounted on the cantilever which interacts with the sample, and we have laser. The laser will be deflected off the cantilever upon probe sample interaction, and we have the photodetector which captures the reflected laser to quantify cantilever bending or oscillation, and we have a sample stage which holds the sample, and we have a piezoelectric scanner which can move the sample, and we have the feedback control system which adjusts the tip height to maintain the laser position. Before we learn about the working principle of AFM, let us see some details about cantilever. The cantilever and the tip are usually made of silicon or silicon nitride. The cantilever has very low force constant for enhanced force sensitivity, and the tip is typically conical or pyramidal to ensure the sharpness. The tip radius ranges from 1 to 10 nanometer, which is critical for high resolution imaging. Let us see the working principle of AFM. The sample surface is scanned by a sharp probe tip mounted on a cantilever to measure the forces between the tip and the sample. The forces between the tip and sample surface cause the cantilever to deflect. So, once it is deflecting, the deflection is monitored using a laser beam reflected from the cantilever on a photo detector. And we have a feedback loop system which can adjust the cantilever height to maintain a constant interaction force or deflection. And this movement is recorded to generate a 3D map of sample surface. Let us understand the working principle in detail. When sample and probe are present at distance, attractive forces occurs and probe deflects towards the surface. As the distance between sample and probe decreases, repulsive forces occur and probe deflects away from the surface. From this picture you can understand when tip is in contact with the surface, repulsive regime occurs, and when the tip is pulled toward the surface, attractive regime occurs, and when the tip is far from the surface, there is no deflection. From this animation you can understand the working principle of AFM. You can see here when the tip scans the sample, we can see the laser deflection that can be detected by the detector, and you will get the final topography of your sample. So, the working principle of AFM is similar to this animation. It is going to scan the sample and it is going to give the final topography of a sample. Let us see what are the various forces involved in AFM. The AFM primarily follows Hooke's law for the interpretation of the deflection, and Hooke's law is F equal to minus K delta x, and this delta x is the cantilever deflection and K is the cantilever spring constant. The various forces involved are the van der Waals forces, electrostatic forces and capillary forces, and these van der Waals forces are weak short range forces arising from induced dipole interaction, and these are dominant in non-contact mode. So, the modes of AFM I will explain later. And next one is electrostatic forces. Due to charge difference between the tip and sample, the electrostatic forces plays an important role. And third one is capillary forces. Due to thin layer of water or other liquid on the sample surface in ambient condition. Let us see the difference between AFM and STM. The main advantage of AFM is we can use non-conducting sample, and also we can do the analysis in air and liquid. We can also use the liquid sample. That is the advantage of this AFM. And in AFM we have three modes, that is tapping mode, contact and non-contact mode. So, these are the advantages of AFM when compared to the STM. Let us see the various modes of AFM. AFM has three modes. The first one is contact mode, and second one is non-contact mode, and third one is tapping mode. Let us see the contact mode in detail. In contact mode, the AFM tip stays in continuous physical contact with the sample surface while scanning. The cantilever deflection is proportional to the force between the tip and sample, and it operates in the repulsive force regime of van der Waals forces. The surface topography is determined by the tip's vertical movement. You can see here the tip is in continuous physical contact with the sample. So, you get very high resolution images, and the application is we can use it for imaging hard surfaces or rigid samples, and we get the high resolution measurements of surface topography. As I told earlier, the advantage is high spatial resolution and we can get the direct measurement of surface properties, but the drawback is if a sample is soft, then it can damage your sample, or if the sample is very hard, then it can damage the tip. Let us see the non-contact mode in details. In non-contact mode, the tip oscillates at a small distance, typically between 1 to 10 nanometer from the sample surface without making physical contact. You can see here the tip is not touching the sample, it is above the sample. It operates in the attractive force regime of the van der Waals forces. The cantilever oscillates near its resonance frequency The tip sample distance is constantly adjusted to maintain consistent oscillation. The applications are, we can use it for imaging soft or delicate samples like polymers or biomolecules, and we can use it for working in environments such as vacuum or low humidity condition. The advantages are, it minimizes the sample or tip damage, and it is also suitable for imaging sensitive surfaces. The main disadvantage is lower resolution compared to other modes. The next mode is tapping mode. In the tapping mode, the tip oscillates close to the sample surface, intermittently tapping the surface. You can see here, the tip is not touching the sample, but it oscillates close to the sample surface. By this way, it can achieve the high resolution. In this mode, it alternates between the repulsive and attractive force regimes during each oscillation. The cantilever oscillates at or near its resonance frequency, but with a large amplitude than non-contact mode. And the tip height is adjusted to maintain a constant oscillation amplitude. The applications are mainly useful for imaging the soft and sticky samples like biological samples or polymeric samples, or we can use it for studying the surfaces with heterogeneous mechanical properties. The advantages are, it minimizes the sample damage as well as the tip wear, and it provides the high resolution imaging comparable to contact mode. The disadvantage is, it requires careful optimization of oscillation parameters. Let us see the substrate properties for AFM sample. The substrate must be atomically flat to ensure that it does not interfere with the accurate imaging and measurement of the sample surface topography. So, depends on your sample, you have to select the right substrate. For example, we can use the mica for biomolecules, thin films and nanoparticles, and most of the lab they use the glass for various samples. It is very easy to prepare sample for AFM using glass, and depends on your application, we can go for even silicon substrate if you are working on semiconductor materials. Let us see the applications of AFM. In material science, AFM is useful for mapping the 3D surface and measuring the hardness. In case of nanotechnology, we can use it for imaging nanoparticles and verifying the patterns and structures. And in semiconductor industry, it will be useful for detecting the defects and irregularities in semiconductor wafers. In biological science, it will be useful for understanding the protein, DNA and measuring the molecular interaction. Let us see how we can use this AFM for imaging the biological sample. So, this is a AFM image of baby hamster kidney cells, that is BHK 21 cells, and this is untreated. In untreated, you can see this is a long cells and this is nanoparticle treated cells. The nanoparticle which is inducing the cell death, that is called apoptosis. You can say in early stage of cell death, the cells started shrinking, and in the late stage the cells become completely rounded and shrinking, and this is an untreated one. And by using the AFM, we can also measure the surface roughness. You can see here the untreated one, the surface is smooth, the cell is intact, so the surface is smooth. Once the cells undergo cell death, that is called apoptosis, the cell membrane is shrinking or blebbing. So, due to that, you can see that the surface roughness is increased. We can use the AFM to measure the surface roughness also. Let us see the advantages and limitations of AFM. AFM provides nanoscale resolution up to 0.1 nanometer, and it is a non-destructive technique, and sample need not to be conductive, unlike other techniques like STM where we need a conductive sample, and also in electron microscopy we need a conductive coatings. And AFM can also measure including topography, adhesion, hardness and friction. It requires minimal sample preparation. The sample preparation is very simple and easy. Some of the limitations are slow scan speed, and it can analyze small sample areas, usually less than 100 micrometer, and it requires a substrate with an ultra flat surface, and it provides two-dimensional topographical data. So, these are some of the limitations of AFM. Let us see some of the challenges in AFM imaging and how to overcome those challenges. The first one is, if you are facing artifacts in images, it may be due to sample contamination or tip contamination or improper scanning parameters. So, clean the tip properly and ensure that there is no contamination in the sample, and also recheck the scanning parameters. The next problem is sample deformation. It may be due to excessive contact force or inappropriate scanning mode. How we can overcome that? We can use non-contact or tapping mode for sensitive samples, or we can reduce the contact force and use softer cantilevers. The third problem is noise and drift. If you are facing this noise and drift, it may be due to environmental factors like vibrations, temperature fluctuation, or it may be due to electronic noise. How to overcome that? We can provide vibration isolated environment for instrument, and if you are getting a poor resolution, it may be due to incorrect tip. Choose the appropriate tip. And the last one is adhesion problems. It may be due to higher humidity, so reduce the humidity or use anti-adhesion coatings on the tip. I hope you got the overall idea about AFM. Let us go to lab and learn this technique more in detail. In this demonstration, we are going to perform atomic force microscopy for a sample of BSE nanoparticles, which we have synthesized earlier in one of the previous lab demonstration. This is an atomic force microscope from Bruker. Here we have two sets of controllers. This one is the stage control unit, which controls the stage movement in the x and y axis. The other one is the nanoscale feedback controller, which controls the vertical movement of the cantilever. The microscope operations are controlled by a software. Coming to the atomic force microscope, the machine is enclosed within a vibration free chamber, which reduces the noise and improves the accuracy of the measurement. This is a stage where we have to mount our sample in the middle. For the measurement, we have the laser source. The laser is generated from the source, which then goes towards the bottom where the cantilever is attached. The laser is reflected from the cantilever and moves towards the photodetector to measure the changes in the tip position with respect to the sample surface. This is a camera which is used to monitor the sample and check the cantilever position. The display unit at the top shows the vertical and horizontal coordinates. Here is the holder in which the cantilever is placed. To attach the cantilever, first carefully remove the laser assembly. The cantilever is then attached at the bottom of the laser assembly. There are multiple types of tips depending on the type of AFM mode. This is the RTSP 300 tip, which we are going to use for the tapping mode. You can see how a cantilever looks. There is also the Scan Assist tip from Bruker, which is a versatile tip used for high resolution imaging of almost all samples. The SEM PIT probe is a multifunctional electrical AFM probe. There are various types of probes available depending on the specific applications. Let us proceed to the microscopy. First, open the software. You can see there are multiple modes of AFM. We will be doing taping mode today. In the setup, select the type of probe that is installed in the machine. The cantilever will automatically come into focus. The small rectangular structure is the cantilever, and the tip is attached at the very end of the cantilever. Now we will align the laser on the top of the cantilever. Similarly, align the laser position at the center of the detector. Now let us load the sample into the machine. This is a BSE nanoparticle sample that has been drop casted on a clean glass slide. Place the slide at the center of the stage and start the process. the sample will move to the center position. Then using the software, first bring the sample surface into focus. Once the sample surface in focus, move the cantilever down till the tip is in focus. When both these steps are done, proceed with the scan. You can see that the sample is being scanned. Then select the desired area to magnify it and take a scan. The raised portions in the scan are visible as bright areas, while the deeper areas are represented in dark color. This is a 3D map of the sample surface that has been captured using atomic force microscopy. The left panel is the 2D height sensor image. This 2D image represents the height profile of the nanoparticles distributed on the surface. The brighter areas corresponds to higher elevations, while the darker region indicates the lower elevations. The nanoparticles appears as distinct, dispersed dots, suggesting a uniform distribution of the BSA nanoparticles. However, there may be a few agglomeration visible as larger clusters at the center and near edges. You can see there are some larger clusters. The right side panel is a 3D topographical image. So this 3D views provides an enhanced visualization of the surface morphology and elevation. The peak represents the nanoparticles, and the height scale in nanometers indicates their approximate size from the measurement scale bars. The nanoparticles exhibit a well-defined spherical or semi-spherical morphology with a surface roughness of around 203 nanometer estimated from this topographical profile. The sharpness and height of the peaks further confirm the presence of nanoparticles on the substrate. As a summary, in today's lecture, we learnt about principle of AFM and various modes of AFM. Also, by practical demonstration, we learned this AFM technique more in detail. Thank you for your kind attention. I will see you in another interesting lecture.